Hello everyone, I'm Jen Hollingsworth and I so appreciate you being here for another episode of the Pathology Practice. I wanted to talk to you today about something that has been floating around the internet. Of course, there's a lot of fear and confusion and a lot of truth to some of these things that we're seeing with regard to the coronavirus, which is actually the virus is SARS-CoV-2, which is the virus that is responsible for COVID-19, the disease that we're seeing in people. And so one of the things that has been passed around has been this whole idea that we are treating the wrong thing. And I think that can come with a little bit of this fear-mongering, but I wanted to help ease your concerns and explain a little bit of the science and the theory behind what we're seeing clinically, as opposed to just this scary thing that doctors are not doing the right thing and other healthcare providers. So sort of the complication that we're seeing with COVID-19 is what ends up being something called acute respiratory distress syndrome, or ARDS. Now normally when somebody has ARDS, it will present with pneumonia on only one side of the lung, not this perfect bilateral ground glass opacity that we're seeing with coronavirus patients. And so clinically, what has come out, and I would say the last week, is instead of patients decompensating as a result of ARDS or pneumonia, what they're seeing is more like this progressive hypoxia. So it almost looks a little bit more like acute altitude sickness. And so I wanted to break down kind of why that might be happening and sort of what are the therapies that are working and maybe aren't the best fit. So again, when a patient goes into sort of, I would say, a position of poor prognosis, usually they're put on a ventilator, and typically we'll see 3 lab values that are elevated in this patient. One happens to be EPO, the second is ALT, which is an enzyme from the liver, and the third is something called D-dimer. And I'll kind of get into that in a moment. But before we get into sort of what does this mean, this hypoxia, I want to refresh us a little bit on sort of the physiology of what we see in the body. And sorry, my pup is in the background. But just the general gas exchange and oxygen and hemoglobin and all those things. So I have a quick diagram to give us a little bit of a review here. Okay, so what I have here is an example of hemoglobin, and it's kind of, unfortunately, we're getting a little bit of light filter for those of you at home. But basically, hemoglobin is what is very important for our red blood cells to transport oxygen. So you have these sort of 4 strands of hemoglobin, or these 4 hemes. In the middle here is iron, and it's a special kind of iron that's sort of caged in what we call porphyrin. Now, what we believe is happening with SARS-CoV-2 is that the glycoprotein of the virus is actually attaching to the porphyrin and releasing that iron. So why is that bad, and what does this mean? Well, as we know, and just kind of again a review on physiology, is that hemoglobin is very important for our ability, for our red blood cells' ability to carry oxygen. So normally you have gas exchange that happens in the lungs, and that little iron flips from an Fe2+ to an Fe3+, so it acquires electron, and then it's in this radical state. But once it's in that state, in the alveoli of our lungs, then oxygen is able to attach, and then as the red blood cells kind of carried out to the rest of the peripheral of our body. Now, what happens with coronavirus, we believe, and again, this is data that's out, research that's out, but not fully peer-reviewed, because we just don't have not had enough time and enough data yet to fully support this. But in essence, what's happening is, again, the virus attaches to the hemi, and we believe the 1 beta strand, and when that does, it sort of like opens up the porphyrin cage, and it allows this iron to just free flow in our body. So why is that bad? One, we have all this oxidative stress because of these free-floating iron molecules. And the other problem is that our hemoglobin is no longer able to carry oxygen. So what happens and what we're seeing again clinically is it's not an issue that our lungs get so weak that they can't pump and we need to be on a ventilator, but that the body is desaturating from oxygen. And we have some lab values that tell us that that's probably a likely story. One, when we don't have enough oxygen, then the kidneys release EPO, and that is so that we can make more red blood cells, because red blood cells carry hemoglobin, and then we're able to attach the oxygen. So now we have basically these Freightliner trucks that, cargo trucks basically, that are moving all around in our body, but they have no ability to carry cargo, the vital cargo that keeps us alive. So, very problematic. The second thing is, again, we have all this iron kind of floating around, and that's why we're starting to see this oxidative damage, the tissue damage in the epithelial of the lungs, as well as and our liver will release something called ALT, because our livers are, our storage for the iron. And unfortunately, if it has more than its capacity, it starts to release this enzyme saying like, hey, I'm damaged, like something needs to change here. Which is the second thing. So we have EPO, which is elevated, which points to poor prognosis. Secondary, we have elevated levels of ALT, alanine aminotransferase. And then third, the D-dimer. So these are all as a result of the body sustaining injury, because this virus has hijacked that porphyrin, and just now floating around on the red blood cell. And again, as we know, viruses are not actually alive. In order for them to thrive and to reproduce, they actually have to go into a cell and hijack the machinery of the cell, so trick the cell into producing more of them. So a virus is either active or inactive. Now, a lot of us have heard kind of this notion about hydroxychloroquine. So why is that helpful? Now, interestingly enough, hydroxychloroquine primarily was used for malaria. It also has other uses for things like lupus, inflammatory disorders, arthritis. But why it worked for malaria is actually kind of similar to why it might be working for SARS-CoV-2. So basically, the reason we end up getting sick from malaria is we have something that's neither a virus nor a bacteria. It's something called a plasmoid. And essentially this plasmoid of malaria goes in, and guess what? It attacks, yes, the hemoglobin. It basically eats away at the hemoglobin. Again, it destroys the red blood cells. We have no way to carry oxygen, and end up with a poor prognosis. But what hydroxychloroquine does is basically inhibits the virus, and in the case of malaria, the plasmoid, from sort of getting infiltrating into that hemoglobin. So the opportunity is very exciting for the continued research of hydroxychloroquine as one of the treatment methods. The other thing we can think about is potentially doing a blood transfusion, especially if somebody has rapidly decompensated and it seems like other therapies are not working. Because what you want to do is, as viral load is going down, you want to restore hemoglobin back into the body. So if you're pumping in fresh red blood cells with hemoglobin, then hopefully the body can reoxygenate, instead of, eventually putting somebody on a ventilator, especially at what's called high PEEP, which can end up causing some scar tissue damage, other things that we don't necessarily like. And unfortunately right now with COVID-19, it's about a 56%, and those numbers are changing slightly, that once we put somebody on a ventilator, unfortunately, many of them do not come off. So the idea is to treat the dysfunction versus the symptom. So again, if we can look at how do we inhibit the virus from attaching to the hemoglobin, which is causing this release of iron and the whole cascade of events, that's where we should look at tackling. Obviously, antivirals are really exciting. There's great promise in remdesivir, and then hydroxychloroquine could be very interesting for its ability to inhibit the virus from attaching to that porphyrin, that cage around the iron on the hemoglobin. And so hopefully that can ease some of your fears and ideally provide a little bit of a different explanation on what's actually happening and how we're learning through this novel coronavirus. Again, this is Jen Hollingsworth. I so appreciate your time, and until next time, be well.